Welcome to Neural Frontiers. Today we are examining the unique challenges of testing neuroscience drugs in humans and why traditional clinical trial paradigms may be inadequate for brain disorders. Neuroscience clinical trials have notoriously high failure rates, over 90% for Alzheimer's drugs alone. Yet these failures often show biological activity without clinical benefits. Are we measuring the wrong endpoints or are we fundamentally misunderstanding what constitutes success in neuroscience therapeutics? Should biological activity be considered a success even without symptomatic improvement?
That's a key challenge in neuroscience trials. We often see drugs showing biological effects like clearing plagiarism in Alzheimer's, but no clear improvement in symptoms. It suggests we might be measuring the wrong endpoints or that symptomatic improvement take longer to emerge. Biological activity is a promising sign, but not the whole story. Ultimately, success means improving quality of life. We may need new biomarkers and longer trials to better capture meaningful benefits beyond traditional symptom scores.
Right. So the placebo effect in neuroscience trials can be enormous, sometimes 40-50% response rate in depression and pain studies. This isn't just a statistical nuisance, it represents real neurobiological challenges. And how do we design trials that account for the brain's capacity to heal itself? Should we study the placebo effect, such as therapeutic mechanism, rather than just trying to eliminate it?
The placebo effect in brain disorders is indeed fascinating, and more than just noise, it reflects the brain's remarkable capacity to respond to expectation and context. Rather than just trying to eliminate it, studying the placebo effect might uncover endogenous healing pathways we can harness therapeutically. Some trials now include active placebo arms to understand these mechanisms better. Integrating this knowledge could improve trial design and even lead to novel treatments that boost the brain's natural resilience.
Right. So adaptive trial designs and platform trials offer opportunity to test multiple interventions simultaneously and modify protocols based on interim results. Given the heterogeneity of brain disorders, how could adaptive approaches transform neuroscience drug development? Are we moving towards precision medicine or improved population level treatment?
Yeah, adaptive and platform trials offer exciting flexibility, especially given the heterogeneity of brain disorders. They let us test multiple drugs or combinations in parallel and adjust based on interim data, speeding discovery and reducing patient burden. This approach supports both precision medicine, tailoring treatments to patient subgroups, and improving the treatment for broader populations. So, we are moving towards smarter, more efficient trails that reflect the complexity of neuroscience.
Right. So the time scale of neurodegeneration and psychiatric disorders often exceed patient lifespan, often patent lifespans and commercial timelines. So prevention trials need to run for decades to show meaningful results. How do we create sustainable funding models for ultra long term studies? Should society be investing in 20, 30-year prevention trials?
Neurodegenerative disorders unfold over decades, which indeed challenges typical commercial timelines and patient lifespans. Sustainable funding requires collaboration between public agencies, philanthropy, and industry, thinking beyond profits to public health impact. Society benefits enormously from prevention. Investing in long term trials is a public good that can reduce future care costs and burden. Innovation Innovative financial models like public private partnerships and milestone based funding may help sustain these essential studies.
So, combination therapies targeting multiple pathways simultaneously shows a lot of promise, but create exponential complexity in trial design. How do we efficiently test combinations without requiring massive sample size and prohibitive cost? Are we approaching a point where traditional RCT methodology becomes impractical for complex interventions?
Combination therapies can tackle diseases from multiple angles but create huge complexity in trial design. Traditional randomized controlled trials RCTs quickly become unwieldy with many combinations. New adaptive trial designs, factorial studies, and advanced statistical modeling can test combinations more efficiently. As complexity grows, we'll need to rethink classical trial models and embrace smarter, more dynamic approaches tailored for the brain disorders.
So, the patient reported outcomes in neuroscience can be subjective and variable, yet they often matter more to patients than objective biomarkers. How do we balance regulatory requirements for objective endpoints with patient centered outcomes? Should subjective improvements be considered sufficient for approval if safety is established?
Patient reported outcomes capture what truly matters, how patients feel and function, which sometimes doesn't align neatly with objective biomarkers. So, regulators still require objective endpoints for approval, but there's growing recognition that subjective improvement with demonstrated safety can justify approval in some cases. Incorporating these both kinds of measures in trials and co-developing digital and biological biomarkers will better reflect real-world benefits while meeting regulatory rigor.
Thank you, Jhansi, for being the part of our podcast, Neural Frontiers, and thank you to our listeners for staying hooked to our podcast. For everyone who likes the podcast and wants to hear more of it, do not forget to like, share, and subscribe. Thank you.